Good morning or good afternoon. Hey guys, and welcome to another lesson. Today we're going to be exploring the second part of division called sexual reproduction or meiosis. Last class, if you remember, we were covering asexual reproduction or mitosis, and that was when cells were growing and reproducing. And they reproduced to either reduce their surface area to volume ratio, so they didn't outgrow their membranes, or they reproduced in order to be able to fill in a gap like a cut, or to repair themselves. So remember, all those different checkpoints would make sure that their DNA was all together and it was all right before they divided, so that would fix any mistakes that had happened to their DNA. Unless, of course, they become cancers, which remember we talked about, and that was kind of a scary that was what happened when your cell cycle was not controlled. So that was the bad part of that, right? So today we're going to talk about, so that was mitosis, and that creates two identical daughter cells, right? And that's important because we don't want to mess up their genes. The whole idea was genetic conservation, right? But today we're going to talk about a process called meiosis where we want that genetic diversity, right? So it's important for your cells in your body, most of your cells in your body, to have genetic conservation so that they don't all get all wonky and cancerous and everything. But there are some cells in your body that we do want to have genetic diversity. Why? So if we think back to evolution, genetic diversity gives you variation in your population. And that variation in traits is going to make it so that, let's say that some sort of disease comes along, or let's say some sort of predator comes along that targets the most common trait in that population. Well, if that population doesn't have variation of traits, that predator or that disease is going to wipe them all out. So we want some variation there so that we have some kind of a plan B to fall back on. So what cells in your body need that genetic variation? What cells don't? How do we achieve that genetic variation? And how is that process related to passing on our traits to the next generation? So this is really kind of taking everything that we're learning about cell division and applying it back to that evolution unit. Because remember that fitness in evolution means the ability to survive, to reproduce, and pass your successful traits on to the next generation. That's really what we're kind of looping back around now at this point. We're getting to the end, and we're looping back around to the unit we started with, which is pretty cool. So let's go ahead and dive in. We're going to actually start with the concept of DNA. So just to kind of go back to our macromolecule unit, doing some spiraling back, DNA and RNA are that nucleic acid. They are the fourth kind of really essential macromolecule or organic molecule that makes up your body. And they're like the instructions, the instruction manuals to make your proteins. So DNA is the basis of like all things in life. We know that all living things share a DNA code, and that's how we know we're all related to each other. And of course, we all share, similar things like metabolism and stuff, but DNA is the big one because DNA is the genetic code that codes for all of your proteins, and your proteins make your traits, and your traits make you, right? So DNA is the instruction manual to make you and all of your features. So DNA, when we're talking about cell division, DNA is the most essential thing that we divide. And you'll notice that even though we need to divide organelles and cytoplasm, we really don't often show it. When we show a diagram of cell division, we're really just showing the division of the nuclear information. We see the division of our chromosomes, right? Not the division of our cytoplasm until the very end here in cytokinesis. But even so, I don't see any other organelles being shown on that diagram because they're not nearly as important. Your cell can make some new organelles, but it can't make new DNA very easily. DNA, if you don't have that full instruction manual, a complete copy of that instruction manual getting to each one of your new cells that you're dividing into, well, those cells are not going to make it. You've got to have those genetic instructions. And so why is this important? What are chromosomes? How is DNA basal to everything we're going to talk about? That's what I want to start with today, and then actually move into the process of division. Okay, so DNA, once again, it's the code for all of your proteins. It's constructed out of a bunch of nitrogenous bases, which is what the actual code is made out of. And those bases are surrounded on the outside by a phosphate sugar backbone, which is, if you remember, constructed by dehydration reactions between the phosphate and the sugars, and it's called a phosphodiester bond. If you remember, we talked about that during Unit 2. And so DNA is crucial to the process of division. But when we divide, we're actually not going to divide DNA in its recognizable double helix form. Okay, the double helix is the form that we use DNA in when we want to actually access it and use it to build proteins, but it's not a safe way to transport DNA. If you guys think about it, your valuables, if you were moving from one house to another, you'd want to make sure that you packaged those valuables up and kept them really protected, surround them in bubble wrap and all that stuff. We want to do the same thing to DNA because we want to make sure that not only is the right amount of DNA transferred when we do our cell division, but we also want to make sure that none of it's damaged along the way, right? Because we just went through all those checkpoints to make sure that it was fine. So why would we want to damage it when we're moving it? So DNA, in order to not be damaged, is going to be coiled into chromosomes. And last time I really didn't get into what a chromosome was. I just talked about how it was DNA and genetic information. But here I want to get into a little bit more detail about how these guys, which look nothing like DNA, are actually made out of this double helix. So I'm going to go ahead and show us this quick video, which is really phenomenal. All right, so here we have our DNA floating around in the cytoplasm. There's our double helix, so it's ready to be used. And we're going to want to go ahead and start the process of packaging it up. So we're going to bring in a protein called a histone. The histone protein is just a little ball that the DNA can kind of wind around like string around little balls, right? So you can imagine it's like you're creating a little ball of yarn. And when we wrap DNA around a histone, it makes something called a nucleosome. Literally just, we call it the ball and string model. So we keep winding DNA around these histone balls, making nucleosomes, and then we keep winding the nucleosomes around each other until they form into this really tight thread. You can think of this again like winding up a bunch of yarn into a ball to be used for like knitting or crocheting later, right? So we're winding up this DNA into this really tight yarn. This tight coiled yarn that we're making out of the DNA protects it during its division process. We call this very tight material chromatin. Okay, and as the chromatin winds tighter and tighter, it's going to go ahead and wind into something that we call a chromosome. Okay, so that's what we're talking about when we talk about chromosomes. And then, of course, we divide that chromosomal information. Last time we learned about this, when we perform the process of prophase, metaphase, anaphase, and finally telophase, right? When we split that chromosomal information to make two identical cells. So that's what a chromosome is. Now, let's take a closer look at our chromosomes. So human beings have 23 pairs of chromosomes, meaning we have 46 chromosomes in each of our diploid cells. Okay, that's a lot that I just threw at you. What the heck did I just say? Okay, when we talk about having a full chromosome count, in other words, you have all the chromosomes you need. So every cell produced by mitosis has a full chromosome count, right? Because we made sure to double them before we divided in S phase so that each one would have a proper amount. Okay, so a full chromosome count is what we call diploid. Okay, and the reason we call it diploid is because we have two of each chromosome. So if you guys look here, we have 23 pairs. This is our last pair, our 23rd pair here. It's not numbered, but it's our 23rd pair. We have 23 pairs of chromosomes, okay, and we have two of each chromosome. So we call that diploid, a full chromosome count or diploid. And di means two, ploid just refers to ploidy, which means the number of chromosomes. So two of each chromosome. Anyway, that's what we call a full chromosome count. Why do you have two of each chromosome, and why are there different chromosomes? Why isn't there just one big chromosome? Okay, I'm actually going to answer the second question first. So I told you that DNA is an instruction manual for proteins, but just like most instruction manuals, there's not just one. Usually really complex processes require like a whole set of instruction manuals, right? And that's basically what this is. Each chromosome has a distinct set of instructions. One might be building your eyes and your nose and your facial features. The other one might be building some of your internal organs. The other one might be building some of your limbs, your hands and arms. Right, and so if you think about it, each chromosome is like a different instruction manual that focuses on a different part of your body. Chromosome pairs 1 through 22 are called our autosomes, and our autosomes are the chromosomes that build your body. Okay, so every body structure. The last pair is the 23rd pair. It's your sex chromosomes, and those build your sexual characteristics. So determining whether or not you are phenotypically male or female, or perhaps both, depending on what you get. Okay, so, and I'm not going to talk about what X and Y means right now. We're just going to know that this is our 23rd pair, our sex chromosomes. Okay, so why am I explaining so much about chromosomes? Well, the first question I think I asked was, why do we have two of each? And that's really what I'm trying to get at here. You might be thinking, oh, we have two of each because during the S phase, we copied each one of our chromosomes, and ta-da, we got, two of each, and we're ready to divide. Yay! I know what I'm talking about. And here I am to come along and totally ruin that feeling because I'm going to say, actually, no, that's not what's happening. Okay, so the reason why this diagram and why all diploid cells have two is because you were created by sexual reproduction between two people, and you inherited one of each of your chromosomes from your mom and one of each of your chromosomes from your dad. So let's go ahead and take a look up here at chromosome pair number 1. So this pair, this guy over here, this chromosome number 1 came from dad, okay, and this chromosome number 1 came from mom. Now they're the same instruction manual, okay, so they contain the same instructions for building, let's say, I don't know, your fingernails, okay? So chromosome number 1, yay, fingernail chromosome. Not really, I don't actually know what chromosome number 1 constructs, but that's not the important piece to know. It's the same detail, okay? So the same instructions, but one is a slightly different version than the other. So if you guys remember, we talked about there are different versions of each gene, and those versions are called alleles. I said it was kind of like, in Super Smash Brothers, you've got the red Mario and blue Mario and yellow Mario and all that stuff. They're all Mario, but they all look slightly different. And that's what's happening here. Both your chromosome 1s have the same basic genes or instructions in the same order, but the genes might be slightly different versions or alleles from one another. So you've got your dad's version and your mom's, and that's why when your body makes genes, it uses both. When it makes proteins, it uses both, and that's why you look kind of like a combination of your mom and your dad. Now, some might be more dominant than others, which is why you might favor one parent more than others in certain traits and the other in other traits. But to get one from mom and one from dad, we call these sets, these pairs, we call this homologous chromosomes. Okay, whoops, my arrow is kind of off there. We call this homologous chromosomes. Okay, so chromosomes, one from mom and one from dad. Now, this is, however, not your chromosomes when they are doubled during the S phase. And you're like, what are you talking about? Okay, so I'm going to show you. We're going to go ahead and demonstrate this with, let's say, another chromosome number 1. We're just going to pretend like we're about to get it ready for division. But this time I'm going to go ahead and draw it. So here's chromosome pair number 1. Okay, so and I'm going to draw, here's dad's chromosome number 1, and here's mom's chromosome number 1. Okay, now when you prepare for division during the S phase of the cell cycle, so remember this is when we're preparing to divide, we need to double our DNA. When you're preparing to do this, we're going to go ahead and double it. We're going to actually double dads, we're going to copy dads, and then we're going to copy moms. And what's going to happen is that one of each of these copies is going to be moved to each of the new cells. So when they divide, we're going to take one of mom's and put it over here, and we're going to take one of mom's and put it over here. And then we're going to do the same thing for dad. We're going to take one of dad's and put it over here, and that's one kid. And then we're going to take another dads and put it over here, and that's another kid or cell, whatever you want to think about. So when we're dividing our cells, when we double it during the S phase, this is what our doubles look like. Okay, now these are still homologous chromosomes, right? They're still considered homologous chromosomes. But if we zoom in, I'm going to try to use a different color here. I guess I'll use black. So if we zoom in here, though, and we take a closer look at what's happening, we've got mom's two copies bound together at the middle by something called a centromere. If you guys remember, I talked about the centromere last time is where those little spindle fibers attach. So we have our centromere, and each of these guys right here are actually exact copies of each other. They're basically clones of each other. So during S phase, we copied each one of mom's chromosomes, and we copied chromosome number 1. So now we have 2 identical copies that are just like mom's. We call these identical copies, these are known as sister chromatids. Okay, sister chromatids, because they are exactly identical to each other. They are different from either of these chromosomes or chromatids, which are they're still sister chromatids, even though they come from dad, they're still called sister chromatids. But their information is slightly different from these sister chromatids, just like the original information here in dad's chromosome, homologous chromosome, is slightly different from mom's. Okay, so we would call, if we were comparing dad's chromatids to mom chromatids, we would say that they are non-sister. Let me go ahead and put this in here. If we compared these two here, we would say that these were non-sister chromatids. Okay, so as a quick recap, the chromosomes that you get that have the same basic genes in the same order but slightly different alleles, those are called homologous chromosomes. You get one from dad and one from mom. And then when we clone them during S phase, each one gets cloned, and we call the exact copies sister chromatids. And if we compare chromatids from the moms to the dads, we call those non-sister chromatids. Okay, so, but if we were to refer to both of these together, so both of these, we would still refer to these as a homologous pair. I know it's a little confusing, and we'll go over it a little bit more as we go along, but that's just sort of some of the basic vocabulary we need to understand before we move on. So when we were doing mitosis, what we were doing essentially, so I'm going to kind of bring it over here, and I'll show it with just this one chromosome, but remember we're dividing all 23 when we do this. Okay, so let's pretend like I doubled it during the S phase, all right, and I'm about to undergo mitosis. So mitosis is going to be the process by which I split this original cell, okay, this original cell into 2 different cells. And essentially what I'm doing when I do that is I am splitting up the homologous chromosomes, right? So here I'll go ahead and show, so we have these guys over here, going to get one from dad and one from mom. And so I basically just split up the homologous chromosomes, right? So basically I've just taken these chromatids and sliced them in half on each one, and I've put the homologous chromosomes together, and I've created 2 new sets of homologous chromosomes, right? So over here, I'll put homologous chromosomes, mitosis equals splitting up homologous chromosomes, okay? The process we're going to talk about today is in meiosis is going to be splitting up the homologous chromosomes and then splitting up the sister chromatids. So that's why I taught you all of that vocabulary. Let's go ahead and dive right in. So if you're confused about any of this, I'll provide some other resources and links for you guys to be able to take a look at. But hopefully, as you go along, it should start to make a lot of sense. Okay, so let's go ahead and start over here with meiosis. So we know mitosis we use for growth and repair, and the idea was to create two identical daughter cells. Okay, where does this happen? So mitosis happens inside of your body cells. Okay, so any cells that are like your skin cells, your hair cells, whatever, those all use mitosis because you don't want your body parts to have a great deal of genetic diversity compared to the other body parts, because like one arm might end up being blue, the other one green, you might end up having 8 toes on your left foot and 10 on your right. That would not be cool. So in order to make sure that every part of your body is the same genetically and it's consistent, we use mitosis or asexual reproduction to go ahead and do that. So where do we want the genetic diversity? We want those in your sex cells. So meiosis is used by all sexually reproducing organisms. That means you and me and anything else that uses sexual reproduction. And meiosis's goal is to go ahead and create genetic diversity so that it can take the alleles, the genes that you have, your unique combination, and shuffle them up and pass them to the next generation all shuffled so that that generation has more variation than yours. Again, the reason for the variation is to make it so that in case the environment changes, your population has a lot of different options to choose from so that selection doesn't just destroy you altogether. Right? Okay, so when we look at the results of meiosis, we should see something immediately different. The results of mitosis were two identical daughter cells. The results of meiosis are four daughter cells. They are not identical. Look closely at these chromosomes. They are different chromosomally. This one has a mostly red chromosome with a blue cap, and the other one's blue. This one has a red cap on a mostly blue chromosome. The other one's red, right? And then these guys over here, this one has a large red one and a small blue one, and this one has a large blue one and a small red one. So all of those cards, those genetic alleles, those chromosomes have been shuffled and dealt differently in each one of our sex cells. The other thing to notice is that they are haploid. What does that mean? Okay, so going back to this, when we said that this was diploid, we have two of each. Inside of our sex cells, we're only going to have one of each. And you're like, okay, at this point you lost me. What is happening? Why do we want a haploid cell count? Okay, so I'm going to go ahead. I'm going to define haploid over, well, actually, you know what? I'm going to define haploid over here. So haploid is going to equal a half chromosome count. So in other words, one of each chromosome. And if we were to picture what that would look like, it would be like we have chromosome 1, chromosome 2, chromosome 3, chromosome 4, etc., all the way through our sex chromosomes. But in each case, we would only have one chromosome. And I'm actually going to make it a neutral color because it could be either, well, I'll do it this way. It could be either moms or dads that you're passing along. It's shuffled as well, right? So you're going to have one of each. Now, why do you ever want a haploid count? Because remember, I said that we needed a full set of instructions in order to be able to run a healthy cell. Well, your sex cells can kind of break some of the rules because remember, your sex cells aren't going to be living independently of your body. They're going to be combined, hopefully, with somebody else's sex cells to produce a new offspring. Okay, and so let's go ahead and consider that. So if we have a sex cell, all right, so we have a sex cell over here, and we call sex cells, another name for them is a gamete. So this is a gamete or a sex cell. If we have a gamete or a sex cell over here, it's going to combine with another gamete or sex cell, right, over, whoops, whoa, whew, that was a big sex cell right there. Okay, there we go. It's going to combine with another gamete or sex cell, and the product of that is going to be a body cell, right? And from what we learned last time, this is going to be, this is going to be a somatic, which is another name for it, or a body cell. So if we're going to make a body cell out of two sex cells, that body cell we know in people has to have a diploid count. So I'll go ahead and say this has got to be diploid. And for it to be diploid in people, if we're going to have 23 pairs of chromosomes, that means that it needs to have 46 chromosomes. Okay? Now, if it needs to have 46 chromosomes, we cannot have diploid sex cells, because that would mean, if we had diploid sex cells, that would mean we would have 46 here and 46 here. And if we add 46 plus 46, that's going to give us 92 sex cells. That is way too many. So we do not want that. So in order to be able to have a proper, number of our chromosomes in our new body cell that we're making, the new kid, we want to have a haploid or half number, 23 of each chromosome, so that when we create the new kid has 46. Okay, so that's our goal in meiosis, is to produce haploid sex cells that are genetically diverse, so they have shuffled genes, to make a unique body cell of a new child that will grow into a new generation with variation that's different than the old and better chances at survival. There's that evolution piece coming back. So we need the daughter cells, again, to have half the number, or a haploid count, and genetic diversity within them. So how do we achieve this through meiosis? Okay, so meiosis, we already kind of went over that, but here's a slide for it just to summarize, right? One allele of each gene, different combinations of alleles, right? Genetic diversity, one copy of each chromosome, 4 haploid cells, and there we call them gametes. Okay, so hopefully that makes sense. Okay, so we actually, in meiosis, we're basically going to do mitosis twice. That's how I like to think about it. Okay, so you'll notice that the names are the same. It's going to go through prophase, metaphase, anaphase, and telophase. It's just going to go through them twice. So PMAT once and then PMAT twice. And when we do this, our meiosis 1 is going to be preceded by S phase. So here we're going to replicate our DNA, and we're actually going to start with a diploid cell that's going to become haploid gametes. So we start with the diploid cell, and that diploid cell is going to go ahead and have homologous pairs of each of its chromosomes, and it's going to have copies of each of the chromosomes, so it's going to have those sister chromatids. And because we have 4 sister chromatids, right, and just to remind you of why, here we can see we have 4 sister chromatids, even though we have 1 homologous pair. Okay, so we have 4 of these. Because we have 4 of those, we call them tetrads. Okay, so we're going to go ahead and we're going to start off with these tetrads. And during prophase 1, we're going to have our chromosomes start to condense and prepare for division, and the nucleus is going to start to go away just like it did in mitosis. But this time, we're going to have a process called crossing over occur. Okay, so how do we actually make sure that each one of the new cells is going to have a distinct or shuffled amount of alleles? Well, that happens during crossing over. So crossing over occurs during prophase 1, and it's where the 2 homologous chromosomes that have already been copied in S phase are going to swap a little bit of that information, that genetic information. So a little bit of dad's homologous chromosomes are going to swap over mom's going to swap over dad's, and they're going to exchange a little piece of genetic information. And this is essentially shuffling their alleles so that we have a new and unique combination of alleles going over. So that's called crossing over. We basically line up our replicated homologous chromosomes. We form what's called a chiasmata. It's just a little twist in the chromosomes, and then we end up with what we call recombinant chromatids, because they have been recombined into new and novel combinations. So that's pretty cool. Okay, so that happens during prophase 1, and so we shuffle our alleles. The next step here is metaphase. Okay, so metaphase 1. So here we see metaphase 1 happening. We're going to line up our tetrads at that equatorial or mitotic plate, right? Excuse me, equatorial or metaphase plate. So we're going to line up our tetrads down there. You can see them all lined up and prepare them for division. We're going to stretch those spindle fibers out to grab them and pull them apart from each other. Okay, and that's going to happen in anaphase. And then, of course, during telophase 1, we're going to get 2 new cells. So interestingly enough, the first division, the first round, we're going to end up with 2 cells just like in mitosis. But then we're going to do something a little bit different. So we ran PMAT just like we did in mitosis. We ended up with 2 new cells. The difference now, though, is that crossing over has occurred, and these 2 new cells are genetically distinct. Okay. Now, there's something else that happens to help them shuffle a little bit. So when we said here that they're going to get genetically distinct, there's something else that happens besides crossing over, and that's the law of independent assortment. Okay, so an independent assortment, what this means is that when we line up our chromosomes for metaphase, what I just showed you, when we line up our tetrads for metaphase, it's randomized as to which side of the cell or which of the 2 new cells created by this process actually receives which chromosome. And so it's totally random. So you can see here that during metaphase 1, we've lined up our tetrads. Here we have a capital R representing this homologous chromosome, a lowercase r representing this one, a big Y and a small y. And we can see that over here, this cell gets the big R's and the big Y's, and this one gets the small R's and the small Y's. But down here, it could also be that we mix those up so that these get the big R's and the small Y's, and these get the little R's and the big Y's. It doesn't matter, but this is just another way of making sure that each one of the new cells that we're creating has a different genetic composition. And this genetic composition is really important. Now, this, again, only happens in meiosis. It's not a mitosis thing, and the idea is to keep shuffling those alleles, those cards, to make sure that each new offspring is distinct. Okay, so we've gone through the first round of PMAT. We call that the reduction phase. We've reduced the number of chromosomes in each one of these cells, and now we're going to go ahead and reduce them again. So DNA is actually not replicated before we go into another round of division. Now, in a normal somatic mitosis dividing cell, this would be extremely dangerous and indicate cancer, but in our gametes, it's fine. Our gametes are allowed to do this, so they don't double their DNA again because we want a half count. We're just going to go straight into PMAT again without stopping, and we're going to have prophase 2, metaphase 2, anaphase 2, and telophase 2, and the end product is going to be 4 cells, which makes sense, right? Because when we start off with prophase 2, we already have 2 cells. They're the cells we created during the first round of PMAT, and so now we're going to start with those 2 cells, and we're going to go ahead and line up our, now we have our chromatids, right? We're lining up. We separate our chromatids, and each one of the new cells is going to get just 1 copy of each chromosome, and we can see that they're genetically distinct from each other, right? So that's really cool. So during the first round, we separated our homologous chromosomes, just like we did in mitosis. In our second round, we separated our sister chromatids. And so now we have 4 haploid cells, which is pretty cool to think about. Okay, so as a recap of meiosis versus mitosis, in mitosis we produce 2 cells, in meiosis we produce 4. Mitosis is diploid because they're body cells, they're somatic. We need them all to be genetically identical, and they need to have a full set of DNA instructions because we're not combining them with each other. But in meiosis, we need haploid cells. And by the way, N just refers to the number of chromosomes. So 2N is 2 of each chromosome, 1N is 1 of each. In meiosis, we need the opposite. We need 1N or haploid chromosome count because we're going to be combining them with other haploid sex cells. And we need them to be genetically different because they are going to create more variation in the next generation. For mitosis, we're going to use it for growth, repair, and asexual reproduction. So just making sure that we have enough cells in our body to keep living and keep growing, making sure that our DNA is all kosher. But for meiosis, it's going to be strictly used for sexual reproduction. It would be very dangerous if meiotic cells were to start proliferating inside the body, because that would, again, indicate cancer. And in mitosis, we have 1 round of PMAT. In meiosis, we have 2 rounds. So in mitosis, we say we have 4 stages. In meiosis, we have 8. At the end of each, cytokinesis is going to divide the cytoplasm. So in case you're wondering, yes, cytokinesis does still happen, just like in mitosis. So they have a lot of similarities, but different functions based off of what we want to do. And again, the whole goal is to divide that genetic information so that in mitosis, each of the cells has a complete and identical copy. And in meiosis, each of the cells has a half and varied copy. Okay, so that is it for meiosis. Next class, we're going to talk about how this actually translates into inheriting traits and different patterns of inheritance. But for now, I think that's enough to leave you with. Go ahead and go through the simulation of meiosis, explore that a little bit. If you have questions, feel free to email me. I'll put in some helpful videos for you guys, help you understand this process, because meiosis and mitosis can be a bit confusing. Just remember that at the very heart of it, the goal is to make sure that you are, I'm going to say, parceling out or giving out DNA to each of your cells in a way that helps that cell to function. Okay, so you are handing out copies of instruction manuals. To your body cells, you hand out full copies of instruction manuals that help them to do their job and keep you alive. And to your sex cells, you hand out half copies so that they can be combined with copies from someone else and to make new and unique organisms that will be able to hopefully survive in the changing conditions of your environment. All right, guys, enjoy the practice, enjoy the simulation, and I will see you on Friday to start discussing genetics. Have an awesome day.